All living organisms contain cells which require nutrients and oxygen to produce energy. Cells also produce wastes such as urea, uric acid, and carbon dioxide. Hence, there should be a continuous supply of nutrients and removal of waste from the cells. This process of supply and removal is carried out by simple organisms like sponges by allowing water to flow through their body cavities. It is also carried out by complex organisms like humans through a special medium called body fluids that circulate these substances throughout the body. Body fluids include blood, interstitial fluid, and lymph. Blood is the common body fluid used for circulating nutrients and removing waste throughout the body in humans. It is a liquid connective tissue that consists of 55% liquid plasma and 45% formed elements called blood cells. Now, let's see what plasma is made of. Plasma is a straw-colored fluid constituting 90 to 92% water, 6 to 8% proteins, 1.2% nutrients and gases, and 0.8% inorganic salts. The major proteins in plasma include albumins, globulins, and fibrinogens. Each type of protein performs a specific function. For example, albumins maintain the osmotic balance by transporting minerals in and out of the blood. Globulins help in the body's defense mechanism, while fibrinogens are crucial for the clotting of blood. Plasma also contains organic nutrients such as glucose, amino acids, and fats, respiratory gases such as oxygen and carbon dioxide, and inactive clotting factors. There are also traces of inorganic salts such as sodium, calcium, magnesium, and bicarbonate in plasma. The remaining 45% of blood is made up of formed elements that include erythrocytes, leukocytes, and platelets. Erythrocytes, or red blood cells, comprise 99% of formed elements. In adults, they are formed in the bone marrow. Did you know that a cubic millimeter of blood contains around 5 to 5.5 million RBCs? RBCs are biconcave in shape and lack a nucleus in the mature state. Their red color is due to the presence of an iron-containing protein called hemoglobin, which plays a vital role in the transport of the respiratory gases, oxygen and carbon dioxide in the blood. The lifespan of an RBC is around 120 days, after which it is destroyed in the spleen, which is known as the graveyard of RBCs. A low RBC count in the blood can cause anemia in humans. Unlike erythrocytes, leukocytes or white blood cells are colorless and possess a nucleus. There are 5 types of leukocytes. Based on the presence or absence of granules in their cytoplasm, these cells are further grouped as granulocytes and agranulocytes respectively. Neutrophils, eosinophils, and basophils are called granulocytes, while lymphocytes and monocytes are called agranulocytes. The chart displays the composition of different types of WBCs in blood. Each of these cells has a specific function in the defense mechanism of the human body. For example, neutrophils and monocytes act as phagocytic cells. and destroy foreign substances by engulfing them. On the other hand, eosinophils fight infections and are linked to allergic reactions, while basophils, the smallest granulocytes, secrete substances such as histamine and serotonin and are involved in inflammatory reactions. In contrast to the other leukocytes, lymphocytes include T lymphocytes and B lymphocytes and are involved in the immune responses of our body. For example, T lymphocytes attack abnormal or tumor cells, while B lymphocytes produce antibodies against foreign microorganisms. Did you know there are around 4,000 to 11,000 WBCs per cubic millimeter of blood? The third component of formed elements is platelets, which are formed in the bone marrow as tiny cell fragments called thrombocytes. A cubic millimeter of blood contains around 1,50,000 to 4,50,000 platelets. They circulate in the blood for around 7 to 10 days, after which they are destroyed in the spleen and liver. Platelets help in clotting by releasing calcium and clotting factors present in their cytoplasmic granules. Let us see how platelets help the clotting of blood. Clotting or coagulation is a protective mechanism that takes place at an injured site to prevent loss of blood. An injured blood vessel releases certain chemicals, which trigger the activation and accumulation of platelets at the injured site. The activated platelets in turn release other factors required for clotting. These factors, along with thrombokinase and calcium, activate the inactive clotting components of plasma to start a cascade of reactions. These reactions catalyze the conversion of inactive prothrombin to active thrombin, which in turn helps convert inactive soluble fibrinogen into active insoluble fibrin. Fibrins form a mesh in which dead and damaged blood cells are trapped to stop blood loss. The mesh looks reddish-brown and is called a clot or coagulum, which is formed in 3 to 5 minutes. As platelets help clotting, their reduction can result in clotting disorders, causing severe blood loss. Apart from blood, interstitial fluids and lymph are other important body fluids that make up our body. Interstitial fluid or tissue fluid is formed when some quantity of water and water-soluble substances squeeze out from the blood capillaries into the intercellular spaces. Its composition is similar to that of plasma and serves as a medium between blood and cells in the exchange of nutrients, gases, and waste. When this tissue fluid enters the lymphatic capillaries, it is called lymph. Lymph is a colorless body fluid that contains lymphocytes, nutrients, hormones, and proteins. Lymphocytes in the lymph help the body fight microbes such as viruses and bacteria. Lymph also plays a major role in the absorption of fat through lacteals in the intestinal villi, where the absorbed fat is drained into lymphatic vessels. This network of lymphatic vessels that collects lymph fluid forms the lymphatic system. The collected lymph fluid, along with absorbed fats, is drained into the blood at lymph nodes. In this manner, lymph, blood, and interstitial fluid play an important role in the continuous supply of nutrients to cells and the removal of waste from cells. Do you know what your blood group is? Blood in humans can be grouped based on the presence or absence of special proteins called antigens on the surface of the red blood cells or RBCs. Based on these differences, Karl Landsteiner categorized blood groups into the ABO and Rh blood groups. The ABO blood groups are differentiated based on the presence or absence of antigens A and B on the RBCs, while the Rh blood groups are differentiated based on the presence or absence of the Rh antigen on the RBCs. The different ABO blood groups are referred to as blood group A, B, AB, and O. The presence of the Rh antigen in the blood is called Rh positive, and the absence of the same is called Rh negative. Did you know that the Rh antigen is named after the rhesus monkey in which it was first identified? Apart from antigens, anti-A